have it set up so that it goes through the material, where we started with plant and cell architecture. And then through water potential, and then transpiration. And so we go through the whole bit of transpiration, but stop after that. So the phloem translocation that we're covering right now will not be on the exam. So the reason I do that, so today's Friday, so more phloem translocation today, probably a little bit more on Monday. I just want to give you time to learn that before you have to do a test on it. So that's why I put it early. I mean, we've already covered transpiration, but phloem translocation is kind of new stuff, and I want you to take time with that to learn it before you get the exam on it. As far as the exam, it'll be multiple choice and essay. So 2 separate exams, and that you'll need to click on and submit. And it's probably not going to be a full 100 points. I don't do, like, if it falls short of 100 points, I don't just like pull a bunch of questions that I don't really care about and throw them on there to make it 100 points. I just don't do that. So if, we haven't covered a ton of information, but I'm guessing it'll be somewhere between 80 to 100 points in that range. Okay. And more points will likely come from multiple choice than the essay, but I haven't finished making it yet, so I don't know for sure on that. So I can update you on that if you need to know more, probably by Monday. And then we will meet here on Wednesday. I will come a little early, like 20 minutes early probably, for any of those of you who can make it, but and that's just to get you to calm down, slow down, double check everything before you submit, so you don't feel so rushed. Any questions about the exam? Okay, I will post into announcements basically what I just said here in terms of what it's going to cover, because a lot of you aren't here today. So I'll make sure to post that into the announcements. All right, so phloem translocation. Did your experiment work in lab yesterday with dialysis tubing? I see some yes, some no. And it doesn't always, I don't know that we've ever got 100% of it to work, like all the setups. So, but as long as we can get 1 or 2 to work per lab session where everyone can see what's going on, that's the goal. And so we need to talk about that a little bit more today in terms of the theory. I wanted to go back to this idea of how, we talked about how they use those darn aphids. We're having issues in the greenhouse right now with aphids. So, but in this case, they actually used aphids for a good purpose instead of having them attack plants and cause issues. So aphids were being used early on in the experiments to try to figure out what's in the phloem translocation process, what's going on, what's dissolved in the water, and you can see sugars definitely are the main ingredient. So that's what we tend to focus on, but there are other things. And then, so sugars, that's kind of vague because, there's all kinds of different sugars out there. So what kind of sugar are we talking about? Now here you can see the way they have this set up is they've kind of, it's an efficient drawing, but let me just show you. So sucrose is being shown right here. So if you take that and drop it down, sucrose is glucose and fructose bonded together. You're going to need to know that. Okay. So sucrose is one of the sugars that they were saying maybe sucrose is in there. But then these are other sugars also that they were looking into more carefully. So the sugar raffinose is just sucrose with a galactose added to it. And then Stachyos has another galactose added to it, and then Verbascos has another galactose added to it. So that's how you interpret this drawing. But when they used the technology necessary to analyze what types of sugars, the one that won in terms of percentage is sucrose. So there's more sucrose showing up in the phloem translocation fluid than any of the other sugars. And when they started looking at sucrose a little bit more carefully, you know, they have glucose and fructose. It's rather small. It's certainly not as big as Verbascos. So it's a smaller molecule. It's a disaccharide. And we say, I don't know if they have it here. Here's another one, a sugar alcohol, mannitol. And so that they noted also in the phloem. And then some other things that they found were certain amino acids. And so glutamic and glutamine are fairly common. And those are in smaller proportion, you know, smaller quantity, but they're there on a fairly regular basis. And then here's some others, some ureides. And so these are especially if the plant is hanging out with bacteria that fix nitrogen, you tend to see these types of chemicals in the phloem. And so these nitrogen fixing nodules that they mention here, that's the idea that this is a plant that is hanging out with a bacteria. So the bacteria is in its roots. And they actually look like nodules or little tumors on the roots. And so the bacteria are busy fixing nitrogen for them, which is creating, essentially when they fix nitrogen, they're taking dinitrogen from the atmosphere, N2, and they convert it to ammonia. And then as soon as they do that, they usually quickly convert it into some kind of amino acid that's being built, okay? So those are other molecules that they see pretty constantly in the phloem in lower proportions compared to sucrose. But sucrose won. It was like the top sugar in terms of sheer concentration. And they wanted to know, why did the plant adapt that mechanism? They know it's a disaccharide. It's composed of glucose and fructose. And then when you look at it more carefully, it is what's called a non-reducing sugar. What that means is that as it's flowing from point A to point B, from source to sink, it's stable and it's less likely to react with things on the way. And so it's just a nice little packet of, you know, sugar that's rather inert or stable in that sense. And then if you think about the bond between glucose and fructose, cleaving that bond gives you a little bit of energy. And so it has a high free energy of hydrolysis, which is good for the plant. That's a good thing. And then again, it's just a small disaccharide, so it's small, physically small. And so that means it's highly mobile. And so these are the reasons that they propose why sucrose is the dominant sugar that's being transported rather than some other sugar. So here you're looking at reducing sugars. So reducing sugars mean that they can react. So the aldehyde groups in particular, the ketone groups, those can be reactive with other molecules in the phloem, things that they encounter on their travels through the phloem. And so that is not a good characteristic for if you're trying to get sucrose down to, or to a sink, not necessarily down, but to a sink, and get it delivered there and let it do its thing at the sink. So we talked about sources and sinks already. We looked at that on Wednesday, and then you thought about that in lab yesterday. And so I'm not going to go over again those slides, other than to think about this one we didn't go through. So we have to think about, let's think about what's happening in the source. So we have, but you know, typically that's leaves. So think about leaves, and they're doing photosynthesis. So the products of photosynthesis, they like to say, they call them photosynthates, and then they abbreviate it PS. So photosynthase is just basically sugar that's being produced through photosynthesis. And so we go from source to sink, and then now with phloem translocation, we talk about source to sink. And this is different compared to when we talked about transpiration, because transpiration, we said it's unidirectional. It only goes one way. Water comes into the roots, goes up through the plant body and out through the stomata as water vapor. This is a little different. With phloem translocation, we can have movement pretty much any direction. And really, when they look at it and they started to try to figure out how is the plant moving substances through the phloem, they do notice that it kind of depends on how close the source and sink are to each other. That does factor in. It factors in like what is the developmental life cycle stage of the plant. So if the plant is busy making a bunch of apples or maybe apple flowers, apple blossom flowers, that is going to affect how the phloem is translocating the sugar. It also depends on vascular connections. How direct are they? Is it a nice, clean, direct connection, or is it more of a more like having to take the side streets everywhere to get from point A to point B? And then they noticed that different things that happen to the plant can definitely influence pretty strongly phloem translocation. So one example that they would do in the experiments of this would be to prune the plant and watch what would happen. So whenever you prune your plants, whether it be your house plant or if you have an apple orchard, for example, and you want, to promote nice, big, juicy apples, you'll often see the grower pruning their trees, the branches and twigs off the tree. And that is a trick to do in order to manipulate phloem translocation and how the sugar moves from source to the sinks. And so all of this they were playing around with in the lab and doing experiments on. Here they actually labeled CO2 and had the plant bring that in. So they fed CO2 around this leaf, so to speak, and then they watched to see how would the CO2 flow through the plant after it's done photosynthesis. So this gets changed into sugars, but it's still going to be radioactive, so they can track how the sugars are flowing through the body. And what they're trying to show here is that it's kind of hard to see, but you're basically looking down on the plant. That's the point of view. And they're saying that this number 14 leaf is on the top side of the plant body, so to speak. And so you see the leaves that are closer to that leaf that got fed the CO2 is getting more of the sugars. So the darker the green, that means they got more sugar than the leaves down here on the other side of the plant, so on the other side of the main stem. So these plants, these leaves were not getting much of anything, but these were clearly getting it, and they are denoting that by the different colors. So number 1 looks like it was getting the most because it's the darkest green being shown. So they're saying that that leaf had more sugar going to it than any other, but also 3, 4, and 6 were getting a pretty decent share of the sugar. And you can also notice that those leaves are smaller. Those leaves are smaller. Number 1 is the smallest. So number 1 is like a baby. You got to feed the baby to get it to grow up. And so they were, the plant was making sugars through photosynthesis and then sending the sugars preferentially to the baby leaf to really promote its growth, make sure it gets off to a good start, and hopefully will reach maturity. And then leaves 3, 4, and 6 are also fairly young. They look more like teenagers, so they're going to need their fair share as well, and are getting more of the sugars than. So 11 and 9 and 12, you see they're quite lightly colored. those are mature leaves. So they're pretty good on their own. They don't really need sugars from leaf number 14 because they're so good at doing their own photosynthesis. But these younger leaves aren't quite there yet. They're vigorously growing, which is a lot of demand for sugar. Plus, they probably don't do photosynthesis as efficiently. Okay. So they were playing around with all of this and looking at, how the sugars are flowing. How does the plant decide, so to speak, where to put those sugars and what direction does it flow? Is it like, water, it's only unidirectional, and they quickly ruled that out. They're like, no, it can go anywhere, really. It can go up, it can go down, it can go lateral through the vascular tissue. So here, figure B, they're just trying to show, this was a technique that was done to show that the phloem is the site of sugar translocation and that it's all open. There's no membranes that are being crossed in those. And they're showing the radioactive flow there. Okay, so that whole mechanism that we introduced and you played around with in lab, they had to think about several things as they're coming up with a hypothesis. The phloem tissue is very different from xylem tissue. It's vascular tissue, just like xylem is vascular tissue. However, the phloem sieve tube elements are living. They are modified. They don't have all the organelles, but they're living. So you have cytoplasm and some organelles and all the proteins and enzymes in there, ribosomes, on and on and on. And then they could take measurements of how quickly phloem translocation was happening from source to sink. So they had to match up, that was used heavily in looking at how to come up with a hypothesis of what force is driving this movement from source to sink. And then they did note that directions are, the way it flows is every which way. There's no unilateral movement like what we see with transpiration with water movement through the xylem. So they're like, okay, we have a lot to think about here. And then they were even getting down, like, especially in the leaf tissue, they're like, how does those leaf cells, they do photosynthesis, they make their photosynthate, their sugars. Then how does it travel to get to the vein, to get to the phloem? And so they were looking at that, getting it, from a spongy parenchyma cell or a palisade parenchyma cell, and then get it to the sieve tube element. And then you have to think about at the sink tissue, getting the sugars or the photosynthate out of the sieve tube element, out of the vein, and get it to its final destination. And so I like to use the potato plant as an idea because a growing potato tuber is like the target destination. You have to get that sugar there to build a lovely tuber. And then we talked about this already. So we've gone through this. I'm going to go through it one more time with you to think about this bulk flow idea. So it's based on bulk flow, not osmosis. Okay, so the movement through the phloem is a pressure gradient. Bulk flow is delta P over delta X. That's the Poisson equation. And so we have a pressure gradient to think about. And so as they're doing these experiments, they quickly realize that probably the main source of water to develop that hydrostatic pressure is coming from the xylem, which is right nearby, right next door. So that's nice. So here you see maybe the source cell is a spongy parenchyma cell in the leaf. It's doing a bunch of photosynthesis. It's making sugars, which are those red balls. Those start to flow to the companion cell and the sieve tube element. And once they get into the sieve tube element, and now you have to imagine there's more than one spongy parenchyma or, one of the cells in the leaf. They're only showing one for ease of looking at it, but you have to imagine there's a lot of these spongy parenchyma cells perhaps right around that vein. And so there's lots of these cells dumping their sugars into the vein of the sieve tube element up in the source in the leaf tissue. And so you can get quite a fast buildup of sugars in the sieve tube element. And as that's happening, you're playing around with the water potential value, that more solute, more negative psi sub S, which makes your overall water potential more negative. And so then water is gonna flow from somewhere nearby, from less negative water potential, which is in this case coming from xylem, less negative, -0.8 to more negative, -1.1. And so getting the water in there, you think about the laws of osmosis, yes. And the laws of water potential, less negative to more negative. And then as water rushes, more and more that water's coming into the sieve tube element, you start to get that hydrostatic pressure. So your size of P value goes up. Here you see it's gone up to 0.6 megapascals. Okay, well, that's half the story. We still have to talk about the other half of the story to have a complete picture. And so all of this is happening up in the leaf tissue. And you need to think about, okay, well, that sugar needs to get down somewhere to the sink. And in this case, when I say down, I'm already imagining the potato plant that has the tuber in the soil. So maybe that sink cell is a potato tuber that's starting to grow and build tuber tissue. So you have to get the sugar out of the vein at the sink end and get it transferred over to that developing tuber. And so if you have unloading of the sugar out of the sieve tube element, that means your psi sub S starts to become closer to 0. It starts to be less negative because you're losing solutes. They're unloaded out of there. So your psi sub S value, it starts to become less negative. Your overall water potential is a little bit less negative compared to surrounding. And that's what they're showing here with the vein, the xylem in the vein. You can see you have a -0.4 in the phloem, a -0.6 in the xylem. So it's less negative in the phloem to more negative in the xylem. So water starts to flow out. And if you have that happening, then you'll have an impact on psi sub P. Your pressure starts to go down, your hydrostatic pressure. And that finishes the story, because now you've explained how you get a pressure gradient. Higher pressure up in the source region in the leaf, and in this case, lower pressure down where that potato tuber is by the veins there, from source to sink. So as long as you have that gradient maintained, you will have flow of sugar through the phloem from source down to the sink tissue. And then you can see in this case, most of the time, what we see happening is water is emptying out at the sink end, going over to the xylem, and then the xylem water goes into the source to develop that pressure in the source. What do you think? Make sense? Okay. So the next couple slides are in words, what we just went through. I have this posted, so you don't need to write this down, but this is basically what we just said while we were looking at that figure. All right, I just wrote it down in case you like to see it in the written word too. So CCCSTE, that stands for companion cell sieve tube element. They tend to think of that as a complex, as kind of like a unit when they describe this phenomenon. And then, so that's phase 1, that's the first part of the story. And then phase 2 is what's happening at the sink. And as long as you think about both of those, what's going on at the source, what's going on at the sink, and you can show that there's a pressure gradient between the two, then you'll have sugar flow from source to sink based on that pressure gradient. So once the sugar is in the sieve tube element, you're just thinking about bulk flow and a pressure gradient. That's all that's driving it. You're not violating the water potential like we talked about. When you look at this image a little bit more, I pointed out Wednesday that you got this weird funkiness with the overall water potential at the source versus the sink. And if you just look at that and think about water potential, you would say, wait a minute, water should actually flow backwards from the sink to the source based on water potential, going from less negative water potential more negative water potential. But that's, when you talk about water potential and water movement with osmosis, that's not happening here. We don't have osmosis happening here. There's no membranes being crossed. Once it's in the sieve tube element, there's nothing there for the water to cross. There's no membrane. And so it's not an osmosis question. Once it's in the sieve tube element, that's not what we're thinking about. It's just the pressure gradient. Okay, so we're not violating any laws of water potential or osmosis. Okay. So once the sugar's in the sieve tube element, it's a pressure gradient you need to think about. So getting the sugars there took some interesting thought and experimentation. You can label the sugars again, you know, the CO2 or the sugars, and actually view it under the confocal. So we have a confocal in our department. Confocals are really nice, those types of microscopes, because you can have living tissue and work with living tissue and run experiments on them, and yet get some really good images. Often when you stain tissue, it kills the tissue, and you get static images, which is good. But if you want to see what's happening with living tissue, a confocal works really, really well. And so here what they did was they did a little surgery on the vein and injected some dye, and we're watching under the confocal the flow, like which way is it going to go. And so that's what a lot of the experiments were early on. They were looking at translocation microscopically and following source to sink movement, trying to figure that all out. Here they've labeled it with the, you know, the green dye, and you can see in the notes section, it explains what some of the other items are in there if you're curious. And then they also did very careful analysis just to make sure that there's no membrane in the sieve plate area. So they were able to show, nope, it's all open from one sieve tube element to the next. It's all open, no membranes. And then one of the questions early on was, is ATP needed? Do we have to do that? Yes. The sieve plate?
Yeah.
In here?
Yeah, it's like.
It's just, what they figured out is like the cell membrane from this sieve tube element kind of goes through. And so you do have some membrane, but it's like a, you know, an open tunnel type thing. And then it's just like, it's all open. So I'm imagining in the sieve plates, the little pores here, you probably have cytoplasm in there, and because it kind of creates like this all, you know, open flow of, there's, you know, cytoplasm and sugars and all of that just flowing. Yeah, so then they were looking at ATP. They're like, hey, you know, does the plant have to expend energy? And which would be rather a bummer if it did, because that would be a lot of energy that it would have to expend in order to get sugar movement. This was an experiment where they were looking at this, and what they knew is that if you take so they took a plant and they had it at, you know, slightly warmer than room temp. And then what they did is they chilled the plant. They put it under cold conditions. And so here, this experiment, when they put the plant at 1 degree Celsius, which would be like refrigerator, a little bit colder than refrigerator temperature. And what they knew is that ATP is sensitive to cold, in the sense that if you chill the tissue like that, ATP doesn't like work. And so they were looking to see if translocation would tank. And initially it did, but that is more the plant just going, ooh. This was a switch, you know, it was a shock to the plant going from 30 to 1. That's shocking. And so this was more of a shock event. And then the plant acclimated and went, oh, yeah, I can deal with this. And it started to do phloem translocation again. If the plant depended on ATP for phloem translocation, it would have tanked and stayed tanked. And so this was just a, I mean, really simple experiment. Let's take a plant and put it into a chilled room. and follow what happens. And that was a really good demonstration that, it doesn't look like ATP is needed, which is good. That's a good thing because I can't imagine the amount of ATP they'd have to generate in order to actually do translocation, especially if you think about, huge trees. So that was a nifty little experiment that they did. Well, then they zoomed into the leaf tissue again, because that's primarily where you have source tissue. And they said, how does the phloem, the sieve tube element, get the sugars? Like what pathway is delivering the sugars into the sieve tube element? And what they noted, if you look carefully at a leaf, it is highly vascularized. So lots and lots of minor veins and capillaries, very much like our blood vessel system. And so they determined that when you look at, many different leaves, they looked at a lot of different leaves, and they said most leaves are only like, there's a 3 to 4 cell distance before you hit a vein, instead of, 10 to 20 cells before you hit a vein. So basically what they were saying is it's highly vascularized. I have a hard time saying that word. So the pathway is short, where you have those spongy parenchyma cells, for example, or the palisade parenchyma cells inside the leaf. The vein is not far from those. And so it's pretty short pathway, pretty efficient in order to dump the sugars into the sieve tube element. Well, when you think about ways of moving things from cell to cell to cell or through the tissue, we've already talked a little bit about this when we looked at root tissue and water movement there. Well, now they wanted to know in the leaf tissue, how do things move? And you have some of the same options. It could be a symplast way, and that is the internal tunnel system where you have the plasmodesmata. And so you just have sugars flowing from cell to cell to cell through the plasmodesmata. And they thought that was a pretty attractive mechanism. And so they were looking very carefully at that. And then they said another really attractive idea would be apoplast again, just like what we see in the root tissue, where you could have sugars moving through the cell wall space of the spongy and parenchyma cells inside the leaf, and then hitting a vein and getting in there. And so they had to look at this. When they looked at the apoplast, they said, well, if sugar goes through the apoplast, through the cell wall spaces, at some point, it's probably going to need ATP to get dumped into the sieve tube. So when it says actively loaded, if you ever hear that in biology, that's intimating energy is needed. And in most cases, it's ATP that we're thinking about. And so they were also looking for that. They wanted to see, does the plant have to use ATP when this is happening in the leaf? Or is it more of a symplast? And so with actively loading, going back to perhaps basic chemistry, I call it the PAT and SAT idea. So PAT is primary active transport and secondary active transport. And this is, I think I have, yeah, I'll show you a little bit more of what I'm talking about there in the next couple slides. So they were investigating all this, looking at all these different plant species, looking at their leaves, looking at how would sugar move, and trying to figure all this out. So let me show you a mock-up here, a drawing of a leaf in cross-section, and you can see the vein tissue. And they're trying to show the darker green cells here. They're saying, let's say those are the ones that are cranking along with photosynthesis, and they have some sugars that they need to deliver to the vein. And so you see the pathway here is what we're thinking about, is going from light green to dark green to the blue. The blue is the sieve tube element. So they're going to take that image and now they're going to blow it up on the next slide here and zoom in on that. So there are your leaf cells doing photosynthesis. And you can see the sugar flow getting to the phloem, well, the companion cell and the sieve tube element over here. So figure A is looking at symplast, where you have Ugh. That internal tunnel, the plasmodesmata. And that seems like a pretty efficient way to do it. And so they were witnessing that when they did the confocal microscope experiments, they could see that. And so it's a thing. And then they said, well, is it happening apoplastically? So here, what you're seeing is, first, initially, it's symplast, but you do see here, they're showing it's crossing foot and then here too. They're saying there's some apoplast movement as well. And so that's where they say if you have, whoops, I gotta go back. Yeah, it's not gonna let me. I can't ever write if it's enlarged, it won't let me. So where's crossing a cell wall and then crossing a cell membrane to get loaded into the companion cell? That's where we have to expend energy to get that to happen. So that is this idea here. So here you're looking at if sugars are in the cell wall space, they have to cross the cell membrane to get in at some point and get loaded finally to the vein. And so here what you're seeing in this mock-up is primary active transport is being shown where ATP is being needed. So they're showing ATP is being cleaved of a phosphate group, and that provides energy to pump a proton across the membrane. So then if you imagine that happening over and over, you get lots of protons building up on this side of the membrane. And then what they're showing is secondary active transport by the movement of a proton coming back in. So now you have a proton gradient. As protons want to diffuse across and even out the concentration, they have to go through a special protein. And as they do that, this flow through there provides enough free energy to get sucrose to come over as well. And so this is kind of like the proton, grabbing hands with sucrose and say, come with me, come across. And so primary active transport sets the stage, get your proton gradient. And then in this case, this proton gradient is being used to drag sucrose across with it back into the cell, or into the cell, I should say. And so this idea of primary and secondary active transport, you have to have ATP being used, proton gradient, but that then sets up the situation where you can get sucrose coming into the companion cell and sieve tube element. So hopefully you've heard of that idea already in some of your other classes, BI 218 and intro biology chemistry, but that's what they were noting the plant was doing if you have the sugar going apoplastically. So they had to look for all of this. It's not just the, they did confocal microscopes, but then they were looking for, do we have these kinds of transporters in the membrane? They had to look for evidence that this is really happening. They came up with these hypotheses, but they had to support it with data. So this slide is just showing that, yes, they found the transporters. So they found the transporters that were allowing for the loading of sugars into the source sieve tube element. So they did find it. So that supported their hypothesis. Here they're showing a leaf, and you can see quite nicely all the vein pattern here, how well vascularized the leaf tissue is. And what they're showing is that, so all the whitish areas really are the leaf cells. And what you're seeing is that sugar, there's so many of the leaf cells all doing this that you get a lot of sugar in the veins. The sugar is the black stuff in the veins, okay? And so you see that you get this great buildup of sugar in the veins by the action of all of that white are the leaf cells doing photosynthesis. So you have to imagine that, you know, you get a lot of sugar coming from different directions and getting dumped into the sieve tube element. And building up that pressure, that hydrostatic pressure in the veins in the source. So they also say here, the sugar moves against its concentration gradient. Okay. So let's think about that a little bit. All right, let's go back to this idea here. All right, here is Simplast. Okay, let's think about this for a second. So if we have many leaf cells, all doing photosynthesis, they start getting the sugar flowing through the Simplast method, so that's through the plasmodesmata, and they get dumped into the sieve tube element. So this, remember, this is all open, you know, this is open pathway from cell to cell to cell. Now imagine where the sieve tube element is here. I think it might make more sense if you imagine lots of, I can't do that. If you imagine lots and lots of other cells, you know, leaf cells, all doing photosynthesis around the sieve tube element, they're all getting the sugars in there, you know, so you got lots of sugar getting dumped into the sieve tube element. So you get at this high concentration of sugar in the sieve tube element with all these open channels. So you have high concentration of sugar here with all of the sugar getting loaded. What's to stop the sugar from just diffusing down its gradient and out of the sieve tube element? So you can imagine where eventually that sieve tube element is going to have a pretty high concentration of sugar compared to surrounding cells. And so why wouldn't it just diffuse out down its concentration gradient? And that was a real thing. I mean, that's the laws of diffusion. And so, and they could see that situation happens a lot where you have lots of sugar in the sieve tube element, a little bit less in surrounding cells. Why doesn't it just flow out of there? And so that was a confusing question that they had to work through. And they came up with this idea called the polymer trapping model. And I remember the first time I read it, when I was learning this process as an undergrad, and I imagined some scientists getting together at happy hour at a local, you know, brewery or pub and having more than one beer and coming up with this one. Because when I first read it, I'm like, what? It was odd sounding, but when you start thinking about it after the initial, like, what are they talking about? It actually starts to make sense. So what they're saying is, okay, you know, by the laws of diffusion, the sugar should go out of the sieve tube element back to some of those source cells down its concentration gradient. We don't see that happening. What is going on? That's defying the laws of diffusion. What they proposed is, they're like, well, okay, what if once it gets into the sieve tube element or maybe the companion cell right next door, what if the plant has a mechanism that it can alter sucrose and add a galactose unit on it to make it bigger? And then it becomes raffinose. And then if you add on another galactose, it becomes stachyose. So they're saying, why don't they just change the sucrose to something else bigger? So polymer is trying to get at that. So sucrose is getting bigger. And then because of that, it's physically trapped. So polymer trapping model. So what they're saying is if you make the sugar big enough, it won't fit back through the plasmodesmata. It won't fit physically. And I remember the first time I'm like, what are you talking about? But really, it does make sense. That plasmodesmata tunnel is only so wide. It's not going to shift how wide it is. It's a certain diameter, and that's that. So if you create a bigger molecule, then it can't physically fit. It's too big. And so that's what they came up with over beers, I'm convinced. And then they said, well, okay, now we got to go prove that. So they went back to the lab, hopefully after they sobered up, and were able to start looking for this idea more carefully. You know, you have to, it's one thing to come up with a hypothesis, but then you have to show that that's what's really happening. So they went back into the leaf tissue, went to the companion cell, went to the sieve tube element, and really did some detailed analysis. So we're talking about this idea, like what we just saw earlier, where instead of just keeping it as sucrose, once it gets loaded into the sieve tube element or companion cell, they just tack on galactose and make it big. And so when they looked for what's going on here more carefully, so the way the artist has drawn this, so what they did was they looked at the diameter of the plasmodesmata, especially coming into the companion cell and sieve tube element, and they could show, so the artist drew this on purpose this way, where the plasmodesmata diameter here between the bundle sheath and companion cell is modest in diameter. And then as you see the sugars, larger sugars, the polymers, getting made and then dumped into the sieve tube element, do you see the difference in the plasmodesmata? And they actually saw that, they could measure that. And they went, hey, this works. Our crazy hypothesis seems to be holding because they do see a larger diameter here. Where you have these polymers able to fit in and go in that direction, get loaded into the sieve tube element, no problem. But now they're too big to flow back out, back into a source cell. And so they propose the polymer trapping model as a way to say that, you know, we always see the sugar going, you know, from one of the leaf cells doing photosynthesis, and it's always going into the sieve tube element. It never seems to back out of it down its diffusion gradient. It just doesn't seem to happen. And so this did explain it. And then they had experimental proof with the diameter, but that they didn't stop there. That wasn't the only thing they looked at. So this is just for study purposes with the extra words for you to look at as you're thinking through this. This is also data to support their hypothesis where they're looking at, or no, is that the one I'm looking for? You don't need to know. There's different kinds of companion cells, by the way. I'm not going to get into that, but they actually could tell the difference. There's different types of companion cells. And so they were able to track what's going on there. And so, okay, so here, okay, this is just apoplastic versus symplastic or passive. And so they were coming up with the data, physical data to support whether or not there's going to be an apoplastic or symplastic method. But where is the, did I take that out? What happened to my other slide? I'm missing a slide. I had another slide in here that was showing. Where is it here? And I'm just missing it. Well, okay, I guess I'm thinking of a different image. So here they're showing that with the symplastic, when they looked at how sugar is going through the symplast, they were able to pick up the evidence that raffinose is definitely there in the companion cell and the sieve tube element, and so is stachyose. So that was showing definitely that you had the polymer idea. So they were able to show that there were polymers and not just sucrose. But there was another image I was thinking of where they got into the enzymes as well. And I don't have that on here. I must have taken it out. Anyways, they did find the enzymes too that are necessary to do that building of the polymers. They were able to find the enzymes in the companion cell and sieve tube element. So it was supporting their kind of wacky idea. But I guess we got to stop there. Are we already out of time? So, okay. So on Monday, we'll get a little bit into unloading I'll touch on that, and then we'll start the next topic on mineral nutrition, okay? Oh, also Monday, I'll leave some time for questions that you might need clarification on for the exam material, okay? So come prepared to ask questions